From Microbe TV, this is Immune, episode number 33, recorded on July 27th, 2020. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi. Hi for live this time. Yay.
Yeah, we're on TV.
In person on Zoom.
We're on TV. I decided, well, the other podcasts are going TV shows, so we might as well too.
Yeah.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here.
And we're all at home.
We are.
We are.
Still.
And it's hot here in Ithaca. I was saying that I'm sweating currently. You might not see that, but
I'm sorry. It's hot. It's either freezing or hot.
Yeah.
In between.
No, we go between summer and winter in like a span of 2 weeks, and then winter to summer in a span of 2 weeks. But there's only 2 seasons. So
yeah, that sounds like upstate New York.
It's 34 Celsius here, which is really warm.
It's warm.
The way I look at it is, we have these 37-degree incubators, which is 98.6, right? So 34 is close. It's in the 90s. Probably the same in Madison, right, Brianne?
Yes, it is 94 here, 94 Fahrenheit.
Hey, somebody's missing. Where's Steph?
Steph had a baby. Yay. Congratulations, Steph.
Nice. Good job.
So I think it was last Monday. Is that right?
That's about right.
Yep. We have another little immuner.
Nice.
Graham.
So now two of my podcast hosts had babies recently, Nels Eldey and now Steph.
Oh.
How about that?
Well, she actually gave birth. His wife gave birth, right. Presumably.
Yeah, Nels did not. His wife gave birth.
So you're getting really the junior team ready to go.
Yeah, because I have to be ready for. I'm not going to be here forever, right? Not to be morbid on the occasion of birth, but congratulations, Steph.
Yeah,
get some rest.
Hey, congratulations.
Pandemic babies, right?
That's right.
Wow. All right. In the meanwhile, we'll continue. If you like what we do, consider supporting us. Microbe.tv/contribute. So we have a very heavy paper today, which Cindy's going to handle. But
I'm going to try.
I just wanted to bring up one paper. You guys will be proud, actually. We just wrote a grant proposal, a SARS-CoV-2, that involves a lot of immunology, a lot of T-cell immunology. We think one of the viral proteins interferes with MHC display.
Oh,
that would make sense. Yep. Mm-hmm.
So we are going to check that out. And so we've been reading a lot about T-cells. So I wanted to tell everyone about there's an interesting literature for SARS-1, right, about immunology. Not huge because, it came and went in less than 2 years, and then it was hard to get grant support to work on it. But a number of people did, including Stanley Perlman. And he has a nice paper in the Journal of Virology called Virus-Specific Memory CD8 T-cells Provide Substantial Protection from Lethal SARS. And this is in mice. It's a mouse model, an ACE2 transgenic mouse. And they say, the antibody response in SARS patients, it was short-lived also. We're starting to see that now with SARS-CoV-2. And so they say here, though, even though the antibody response in people for SARS-1 is short-lived, the T-cell response, CD8 T-cells, can persist up to 6 years after SARS-CoV infection. And at that time, memory B cells and antibodies are undetectable. Undetectable, right? It's amazing, right? Because usually you would think at least memory B cells, low level antibody, but no. But the T cells.
I don't know though. I always think about it as those memory cells. They hang out in places we can't always find them, right? In humans, we sample blood, and that's not where they like to hang out.
Where do they hang out?
Memory B cells can go back to the bone marrow.
Bone marrow, yeah.
And be continuously producing low levels of antibodies, which you may or may not be able to detect because the titers might be really low.
Yeah.
Yeah. So the antibody part of them being undetectable is a little more surprising, especially depending on how they tried to do that detection. Yeah. But the B cells might be harder to find.
Nevertheless, they found peripheral T cells, no problem. And then they did some experiments showing that T cells can protect mice from infection in the absence of antibody. So they say our results show that memory CD8 T cells will protect hosts from lethal infection, but antibodies are probably necessary for complete infection. And the reason I wanted to mention this is, everyone's talking about antibodies, especially in terms of the vaccines. And as Rhiannon and on TWIV, we're always saying, they do these vaccine trials and they do a few here's some T cell, here. And then they say 2 sentences about them and ignore them. And I'm thinking, they're probably important, right?
They are.
For SARS-1, they are. And
they are much harder to assay, though.
Yeah, it's exactly right. And so,
I think that I heard you talking about that, Brianne, on TWIV the one time.
Yeah, and the big problem as well is not only are they harder to assay, they're harder to assay in a rapid way. And so if we wanted to actually look at everyone's immune response in a population, it would be really hard to, see if you have T cell memory.
Right. But I think that, the way I always teach this, the important thing to keep in mind is that there are parts of the immune response that are designed to protect new cells from being infected, right? Those would be antibodies, because those antibodies can neutralize, right? So we talk about neutralizing antibodies. So they can bind the virus and prevent it from infecting a new cell. But that in and of itself isn't going to get rid of the cells that are already infected.
Yeah.
So the T cell arm eliminates the infected cells. And so both of those work complementary together to be able to protect the host. And so, yes, the T cells alone work a little better because they can eliminate any virally infected cells. But if you keep killing the virally infected cells and releasing some more virus here and there, you're going to still have new cells infected. So it's going to take a really long time to eliminate the infection. But if you also have antibodies on that, it's better. But just antibodies alone, it's hard to eliminate an infection that way. So you need both.
Yeah. And assaying neutralizing antibodies is easy. You just do a plaque assay. Whereas if you want to see if they're CD8s that will kill an infected cell, it's harder, right?
It's much more complicated. Because you have to have MHC matches and you have to have the proper antigen presentation and the right T cells. Yeah. So it's definitely trickier to do.
Okay. So with that little background, we have an amazing. It's not just T cells; it's T and B cells, right?
Oh, it's everything.
It's everything.
So I have. You're going to hear pages here. I'm going to be honest with you. I have like 8 pages of notes because this paper is really, really intense, and I'm going to try and break it down into a very simplistic form that I can understand, because I don't do these big data experiments, and there's just so much in this. It's such a rich paper with so much information in it that it's so hard to dig through and get all of that. So you kind of get the surface ideas, but they're going to be analyzing these data for a long time and finding new cool things, because there's just so much to look at. And they just scratched the surface. So the paper that we're going to talk about is called Deep Immune Profiling of COVID-19 Patients Reveals Distinct Immunotypes with Therapeutic Implications. And the main author is John Weary. And the first author is, how do you say this? Devij Matthew?
Yeah, there actually are 7 co-first authors and 3 co-senior authors.
Yeah. Well, it's a big paper. I mean, this is not an endeavor that an immunologist alone could do. You need some serious bioinformatic people, because there were types of analyses in here I haven't heard of before that were really cool. But the main idea was we really don't, and by the way, this got a huge amount of press for this idea of immunotypes. So that kind of got the splash there, because everybody's like, oh, if you could predict which immunotype you are, you might be able to predict your outcome of infection, right? Because that's the holy grail of understanding this disease, right? And so basically what they did was they had 125 either recovered or healthy patients, and they were able to define 3 immunotypes. And so they did a lot of different analyses on T cells, on B cells, on antibodies. And then they just did these amazing analyses of trying to look at these correlations between different biomarkers and different outcomes in different immune cell populations. So we'll go to that. But I thought one of the interesting things that theirs was pretty consistent with having a certain population of individuals that just didn't respond well. And I think that seems to be a recurring theme of many, many different papers these days, where up to 20% of patients are just not having a robust response. And so we wonder what's going on. How are they being protected, and does that mean anything? And I don't know if we still know whether it means anything or not. But
right. And are those patients? We should sort of clarify. About whether those 20% have no response and do really poorly, or have no response and are just fine.
So it seems like they're no response and doing quite fine, yeah. which is weird.
Which is weird. But that's what they're talking about with this group of patients.
That's right.
You can't really measure a big immune response, and those patients aren't getting particularly sick.
Right. So they have these 125 of these recovered individuals, which not presumably. So they've been sick before, but they recover. And then they also have healthies. And then they're going to compare those to 149 individuals who were definitely positive and were hospitalized. So it's a little skewed here because they're only looking at the really severe people, and then people who have recovered. So there isn't really an in-between to know whether those people who were, kind of sick, but then didn't require full ventilation and full on interventive therapy, how they did. But the recovered ones are supposed to be the ones that got sick but stayed home. They didn't need to be hospitalized, and now they're better. So it's a caveat, but it's how they compare these. And they have statistics for how many got different treatments. There was a lot of them that got hydrochloroquine, which we now know is not great. And this actually shows there's not a great correlation. Oh, we can get to that. But so they have some that died and some that didn't. And so the idea was to try and correlate these things. And what was nice was the first figure, they really talk about clinical findings. And so it was good to see that a lot of the things that we've been talking a lot about, they saw. So clinical findings like D dimers, because we've talked about coagulopathies, and C-reactive protein as an inflammatory marker, and lymphopenia. They saw all of those things recurring in the severe patients. So they didn't see a huge correlation with IL-6. There was some of their patients had high IL-6, but then there were some that were normal. So for things when you're thinking about treating with tocilizumab or other anti-IL-6 therapies, it makes you wonder, if these are in these immunotypes, do all of them need to get this kind of therapy? And how, even if they have high IL-6, is that actually an indicator of how their disease is going to progress? And I don't know that we know fully that information yet. But they did this correlation analysis with disease severity. So they use these Apache scores and these NIH correlations, and they have these beautiful figures where they're trying to show you, like, for example, white blood cell count and disease severity or things like this. And a lot of these, I don't know what you guys thought, but some of them were very clear, like there's a clear association, and some of them they say were an association, and I wasn't so clear about it. But I think some of the more significant ones were white blood cell count. So when they have lymphopenia, when they have elevated neutrophils, they correlated with disease severity. And things that we've heard a lot about: age, mortality correlated with disease severity, and things like metabolic disease and things like that. They also saw correlations with those. So that's good. But then, so they show these correlations, which some of which were expected, and some of them, they popped out a little bit more. But then they started doing the really cool stuff, and they took peripheral blood mononuclear cells from these patients, and they did this high dimensional flow cytometry. And so this blows my mind because when I was a grad student, we had like one color and it was like really amazing to do like 2 or 3 colors and you compensate. And it was like, whoa, they're looking at like 39 different parameters or 50 different parameters, plus then looking at all these different cell populations and all the different activation markers that they're expressing and all the cytokines that they're making. And so there's just this incredible power that they can analyze with all of this dimensionality to these immune subsets. And so
I am very glad I didn't have to compensate that data.
I think it's all automated now. You have all of your controls in place and you run the machine and you run all the
What do you guys mean by compensating?
Oh, good question. So for those of you who might do microscopy, this might sound a little familiar, but if you haven't done flow, if you have 2 colors, and the way a flow cytometer works is you label cells and then you run them through a microfluidics device and shoot a laser at them. And if they have 2 colors, the 2 colors will fluoresce in different wavelengths, and then you read that information. And it's great if you have 2 wavelengths that do not overlap at all with their excitation and emission spectra. So you have to excite with one wavelength and they emit in another wavelength. And we tend to think of that as binary, right? They emit in a wavelength, but they don't really emit in one wavelength. They emit in a bell curve over a number of different wavelengths. And so their peak wavelengths where they emit is their strongest signal, but they often have a tail that goes out. And so if you have another fluorophore that will excite close to that, you can have these overlaps. And some of the signal is coming from the other color, not the first color. And so you have 2 colors, you can say, okay, this peak is here and this peak is here. And so you can just not collect the information in between and separate them pretty easily. But the closer and closer they get, the harder and harder it is. And if you add 3 colors or 4 colors or 15 colors or 20 colors, it gets more and more difficult to compensate. So take away that color that's due to the other
bleed over. Yes.
Right.
Yeah. And so when I started graduate school, we would have to run controls and then basically turn up or down certain parameters to try to do that all manually. By the end, we were doing most of it digitally and having the computer figure out what to subtract.
Right.
You may not remember this, but when I was a student, we had scintillation counters, right? And you could measure isotope emission in them. And you could measure tritium and C14 and P32 and S35, and they all had bell curve and they all had overlaps. And we had to do the same thing. We had to count individual samples and then see how much they bleed over and subtract that. Of course, back then we had to do it manually because you didn't have any software. It's the same idea. It's amazing.
It's the same idea. Yep.
Cool.
Yeah. So they did this high dimensional flow cytometry. So the amount of overlap with this many different colors is just insane. But what they started to see was different changes in cell populations. So they saw decreases in B cells, decreases in CD3 positive T cells. So we know CD3 is a marker for T cells. And they saw both CD4 and CD8 losses. And the lower T cells correlated with the increased markers, the other biomarkers like D-dimer and C-reactive protein. So it seemed like the changes in the immune cell populations were also correlated with the increased severity of disease biomarkers. Now, remember that there's CD4 and CD8 T cells, right? So the CD8 T cells are the ones that we were talking about before, where they're the ones that go around and they kill the virally infected cells generally. And the CD4 cells generally are the ones that are helper cells, and they produce cytokines that trigger the B cells to make antibodies. It's simplistic. We could get into a lot more detail there. It's not an exclusive, either or. There's always, exceptions to the rules in immunology as there are everywhere. But generally, that's how we think about it. So if you have a loss of CD4 T cells, you're likely going to have a loss in the ability to generate good antibody responses. And if you have a loss in CD8 T cells, you're going to have a loss in ability to kill virally infected cells. So, yeah.
Yeah. So one other thing from the data that just I happened to notice, I think it was, they don't comment on it, sort of a side point. They actually looked in their correlations at blood type.
Yes, they did.
Which has been in a few different other reports as well. And they really didn't see anything. For example, the biggest thing that did not correlate with having type A blood was having type B blood.
Yep.
And having type O. And those
That's a good thing. Their analysis worked.
Yeah. So those were the only things that actually came out of their blood type analysis.
But that's an interesting story because I happen to be an A blood type, and that was the one that they said, oh, you're going to have more severe disease. So of course that caught my attention, and I started looking into it, and I was like, I'm not sure if I believe those data. And now we're starting to see more and more papers come out that say, no, there's not really a strong correlation there. So that's great.
Good.
Right. So then after they did this general analysis and biomarkers and sort of what looks different, then they started to get into the really interesting stuff. Because they did this high dimensional analysis, they could drill down into specific subsets of cells and ask what's going on. And so the first cell type that they dug down into was the CD8 T cells. And so again, these are the ones that are required for killing virally infected cells. And so they did this, and I'm not going to claim to fully understand all these bioinformatics, but generally the idea is they do what's called a principal coordinate analysis. Mm-hmm. So in this, you ask the data to tell you what drives differences in populations. So if you say, take 193 different parameters that they looked at, and ask them to just group based on healthy or recovered or COVID status, and just, can you separate those in multiple dimensions? And the answer is yes, they cluster, and the recovered and the healthy cluster more, and they cluster away from the COVID, which is comforting. But then you can start to say what drives that difference. Like, what exactly is it about component one or whatever that causes the COVID patients to be able to segregate from the other ones? Which of those 193 things, or which several of those 193 things, cause that separation? And so they started to look at, the T cells and the CD8 T cells, and they define multiple different combinations of T cells. And then they're going to ask, are those different, and do they drive some of that difference, that spread? So then they get into these different T cell populations, which, again, I'm an immunologist, but I'm not a T cell aficionado. But generally, there are what we call naive T cells. So those are the ones that have never seen antigen. So they're just flying around the body looking for trouble, looking for something that they can go and then fight, right?
They're not yet activated.
They're not yet activated. But then we also have activated T cells, which are currently fighting something. And then we have memory T cells. And memory T cell immunologists love to subsets of subsets. And so these guys looked at central memory cells, and then effector memory cells. And Brian, you might be able to speak to this a little bit more than I, but the effector memory cells are the ones that are just basically poised and ready to go the next time you come into, the infection. Whereas the central memories are sort of reserving some level of plasticity so that when that infection comes again, they can go down different potential paths.
Yeah. So this is related to things I was doing as a graduate student, although the number of different subsets in this paper was a few more than I remember spending time thinking about. But I usually think of the effector memory cells, just as Cindy said, in that they are sort of poised to be able to respond again. And they also are usually in the anatomic location where infection was previously seen. And the central memory cells are adapted to be long-lived. They proliferate a little bit better, and they tend to be sort of stored away in lymphoid organs.
Yeah. So they took these effector memory cells and subdivided them into 4 groups, which I had heard of 2 before, but now there are 4, and it was more than I knew of.
Because
and I'm
they
not
can.
Sure.
Because they can, right?
Yes.
Okay.
Yes,
yeah.
Because they can. So they can stain them with multiple markers. And once they compensate, all of a sudden there's like slight differences in where these guys appear on the dot plots. And so they put circles around them and they call them new new populations. The important thing is, do they have a different function, right? Because it's nice if they have a different marker, but they seem to say they do sort of have slightly different functions, and so we should keep that in mind. But basically, some of the things that they were seeing drive this difference, this coordinates difference, is there's a reduction in one of these effector subsets called EM1 and an increase in two other ones called EM2 and EMRA. So what does that really mean? It just means that there are differences in the effector memory populations that these COVID patients, maybe they have different levels of memory that have developed at this point. So the other thing we have to keep in mind is we're measuring memory cells in somebody who's still currently sick. And so I think that what they're really saying is that the ones that have already recovered have more of these effector memory cells, which makes perfect sense because they've already recovered. So that could be driving a lot of this difference. But one of the other things they looked at were some markers of exhaustion or of just the T cells have just gone overboard and they are now shutting down. And so this is kind of interesting because if they have these markers of exhaustion, it means that maybe they're no longer functioning in the body. So they upregulated these markers that say, I'm overactivated, I need to quiet down. But if the infection is still ongoing, that's not really what you want to happen. And so that could actually be important. And so they did see an increase in some of these markers associated with some of these populations.
Yeah, and I think that is a really important piece to this because they talk in a few places about how there have been different hypotheses about how severe COVID disease might be due to your immune response being too low or your immune response being too high. Immunologists love Goldilocks. And the fact that there is increased exhaustion in the T cells is support for the idea that perhaps the immune system being too high is a problem in these patients. Right. That excess immune response could be leading to exhaustion.
Right. So there's also a couple of other things that they look at related to these different T cell functions is proliferation. So they had more proliferating cells, which, some of these things you're like, oh, that's cool. And then you're like, wait a minute, that makes perfect sense, right? They're sick right now. So their T cells are proliferating to fight the infection. And the ones that are better shouldn't have that happening anymore because there's no more antigen around that's driving the proliferation. So they see that, and they see fewer naive T cells. So again, that makes sense because they're currently fighting the infection. But what was interesting is that it comes back to this non-responders, right? So they had 20%, and I think that's a kind of a high number, of their patients that did not have any proliferate or had very low levels of proliferating T cells. And that's surprising. And that's suggesting that the T cells are not functioning properly. And that could be a really important biomarker.
Which could be why they're sick, right?
Right, exactly. So if their T cells are not working properly, then you would be more sick from more virus replicating, right?
You think that's a high percentage, or is it just because the population is biased? These are people who are in the hospital, basically, right?
I mean, if you're more than a week out from the initiation of infection and you don't have any T cells, I think that's somewhat surprising.
A problem.
Yeah.
And it's also sort of hard to tell from this. They look at sort of what percent T cells are responding, and they sort of make some comments about whether those are just the T cells responding to COVID or whether this is broad T cell activation, which again could be happening if there were excess cytokines, maybe from innate immune responses or something.
Right.
And they sort of say, well, it could be either, and we don't really know. They don't completely answer that well. I spent some time staring at these data, trying to decide what I thought about the percentages of proliferating cells, and I'm not sure I had a great answer.
Yeah, I didn't either. I think without doing a detailed analysis of antigen specificity, you can't really say.
No.
Right. I mean, because we can have antigen specific T cells, which is what you want. But then sometimes if an infection or inflammation is so strong and there are so many cytokines around, you get this bystander activation of T cells that shouldn't happen, quote unquote. But maybe it's just a self-preservation mechanism. We're going to try and activate anything we've got in case there's something there that can fight what we're not winning against. But it's not a typical outcome, I don't think. So I thought they did all of these interesting analyses that are kind of cool. So for those immunologists who are not familiar with all these bioinformatics, we often hear of Tisney plots. And I have to admit that when I very first heard someone said that, I thought they were calling it a Disney plot because it's multicolored. But it's a Tisney plot. And what this does is basically similar to that principal coordinate analysis, but you're asking the cells to cluster according to gene expression or whatever parameters you're looking at. And you get this multicolored plot, and then they draw bioinformatically circles around all these different populations. But what's cool about this is then you can interrogate your gene of interest and ask which cells express that or your marker of interest. And so what they were able to show is that there are increases in certain populations that are associated with disease, and they have higher levels of certain markers. So generally you have lower T cell proliferation, but there's this one subset that's higher T cell proliferation, but it also, that same subset has higher of these markers of exhaustion. So it's sort of saying, like there's the expansion of this one population that probably would be helpful, but it's also being shut down at the same time. So you can look at which populations overlap these markers. I don't know if you wanted to add anything to that. I was trying to be somewhat general in how we describe these really complicated data.
Yeah, I think that makes sense to me. So, when I look at some of these figures, I'll say, oh, it looks like there is a group of cells that happen to have CD27 and TCF1 in common.
Right.
Or another group of cells that has TBET and CX3CR1 in common. And so it helps us sort of understand the subgroups of types of cells in a little bit more dimension.
Yeah, and I think it's more unbiased, right?
Right.
So when immunologists tend to subsets, we pick our favorite marker and then we co-stain and we see if it's on there or not. And then we say, or, not. But what this is doing is we measured a whole bunch of things at once. I didn't, but they measured a whole bunch of things at once. And then they ask them to cluster these cells, which ones express multiple things. And then, so how can we learn something about that population? And honestly, I don't know enough about all these different markers to be able to say anything more than they were saying that, that seems to be that the ones that you want to be fighting the infection are the ones that seem to be suppressed in some way. So one other thing that they did was they showed that there was increases not only in just like markers of exhaustion, but also adenosine generation, so the CD39. And so that's a soluble suppressive cytokine. So it might be shutting down some of these responses as well, which could be interesting. So that's the CD8 T cells. And then they basically photocopied that onto CD4 cells and did the same thing and asked, were there any differences? So I started out like really excited about this paper. And then I was like, oh, they're just going to do the same thing on cell population after cell population. But, what did they find? They found there's a decrease in the naive CD4 T cells. So that's expected. And then they had these memory cell populations and they saw very similar changes in, up or down these memory cell populations in the disease. But what we have in the CD4 T cell population that you don't have in the CD8 T cell population is this TFH, or T follicular helper T cell. And so that typically is located in a lymph node. It's interesting that they find those circulating around, but they tend to be the ones that are helping the B cells make antibodies. So they're indicative, if you have a high level of these, that's indicative of a good antibody response ongoing. And so they did see increased numbers of activated T follicular helper cells in the blood of the COVID-19 patients. But what was, I thought, interesting is they also saw these remaining high in individuals who had recovered. And so does that mean they had some level of ongoing low-level infection, or that there's some sort of mechanism to have that antibody response persist, even though we talked about antibody response is sort of waning, but the T follicular helper cell response seems to be still high. So it's a dichotomy that I can't say I totally fully understand, but I think it is unusual to have an extended persisting T follicular helper cell response measurable following a viral infection.
Yeah, and I think that really highlights a place where there's more research needed in terms of trying to understand the fact that we see this persistent T follicular helper subset and perhaps a decrease in antibody, and how do those things shouldn't go together, but nope. They're happening at the same time. And so what exactly is going on? This might sort of point to something new that should be examined.
Agreed. Yeah. I mean, you can't, these type of papers, you can't argue with the data. They are what they are, right? I mean, they just do this analysis and then just let it happen. That being said, the bioinformatics is not tricky, and two different people doing the analysis could come up with two different things. And so we need to wait and see whether these things are reproducible and whether they look at these in more patients without doing the high dimensionality, but focus in on certain specific subsets without all of the other staining. Does this really hold true for many, many individuals?
Yeah.
So I think we need to wait on that, but it's still intriguing that there might be something going on. My first thought was, if these T follicular helper cells are circulating, well, then they're not where they need to be doing their job helping the B cells. So maybe there's something about, and they did mention something about some of the chemokine receptors which they express. Maybe they're downregulating the ones that are necessary to stay in the lymph node, and so then they're circulating when they shouldn't be, and maybe that's somehow affecting the antibody response. But I think there's, like you said, more we need to do to understand that.
I hadn't even thought about that. The idea that the T follicular helpers were lost.
They're just wandering around aimlessly in the body. I don't know, where's my B cell that I need to be talking to, but yeah.
I always look at big data like this as sort of a great way to come up with more questions, more specific questions to then nail down on.
Yeah. The other thing I thought that was really interesting was they started looking at, this Ki-67, so this proliferation marker again, and this time in CD4 T cells and not CD8 T cells. We're done with the CDAs. But so 2/3 of the COVID patients had Ki-67 staining in non-naive T cells. That means they're reacting. So that's good. Their T cells are doing what they're supposed to be doing. They're proliferating. They're then going and helping B cells, hopefully, if they're not lost. But 1/3, that seems like a really high number to me. 1/3 did not have proliferating CD4 or CD8 T cells. So that says that they're really having a defect in their T cell response. Now, one could say, is it lagging? Have they just not quite gotten there yet? So, we don't know the kinetics specifically at this point. But it still is somewhat surprising once you're hospitalized, if you're that sick, that you're not having a T cell response yet. So, other things that they look at, there was also, the Disney plot, the Disney plots, and they, again, they found certain populations of cells that co-express certain markers, and they did cluster analysis, and they found these clusters, and they could show that according to disease severity, there were increases or decreases in these clusters of cells or, groups of cells, and, which ones are driving the disease. I don't know, but the takeaway message that I took was that there was a decrease in this circulating T follicular helper cell population that correlated with severity of disease. So, we talked about there being fewer T follicular helpers, more in the recovered, but there seemed to be a few of these activated ones that were more in the disease patients. So again, is it maybe they're lost, they're activated, maybe they're not quite getting where they need to be yet? I don't know. It was really confusing to me why generally you might have fewer TFH cells, but yet you have an increase in this one that's supposed to be active, and yet you're finding them in the blood when they should be in the lymph nodes. So, yeah. I think it's an interesting observation. A lot of this is correlative, descriptive, observational. We really don't know which things might be driving certain parameters, disease parameters, or disease severity at this point.
But as we go forward, and when we get to the sort of specific immunotypes part, then we can sort of come up with some ideas or hypotheses about what we would want to know to figure those things out.
That's right. So the one other thing they did do was they did culture some T cells, which was good. They couldn't do it from all the patients because if you think about this, if they have current SARS infection, that's a BSL-3, and it makes it really tricky to be inducing T cells that, are associated with a viral infection. But they were able to culture some of these, and they were able to show that some things were surprisingly not up, like interferon gamma. I didn't see them talking about type 1 interferon, which is interesting because there's a few paper that suggests that type 1 interferon might be defective in these patients. But they did have markers that were consistent with an inhibitory phenotype, like IL-1 receptor antagonist was up, and CCL2 was up, and that's a monocyte attractant, which fits with their increased monocytes that they see in the blood. So, but they were only able to culture the PCR negative patients. So they're severely ill, but they're no longer PCR positive for disease. So we have to keep that in mind as to, how do we interpret these findings that they found.
And one thing that's really sort of interesting there is they saw that sort of low or lower than expected amount of interferon gamma.
Yes.
I wish that they had also looked at TNF.
Yes.
TNF alpha. And the reason why I say that is that there have been a couple of those vaccine papers that Vincent mentioned that do a little bit of the T cell data. And one of the things that I've noticed in some of those papers is that many of the vaccines are inducing T cells that make a lot of TNF and not a lot of interferon gamma. And so it would be interesting to know if it's similar in these patients with more TNF and less interferon gamma. And
so we technically like to see more interferon gamma, less TNF, because TNF is more pathologic, drives more immunopathology. And interferon gamma is better for fighting intracellular microorganisms. So they measured some things and not other things. Some of it might be buried in their data. Maybe they chose not to, show it. I didn't look and see whether TNF was in their panel, because if it is, I assume they've deposited all of their data. And if one were, bioinformatically savvy, you could go and interrogate those data and find out a lot more information about your markers of interest.
That is true.
Yeah.
I hope one of my students isn't listening right now, so he doesn't know that something we can talk about.
Wait, so that you can bring it up and seem smart, or
yes, exactly.
See, one of the trainees, you don't understand that the PIs are always trying to stay one step ahead of you. So that they're like, "Oh, did you see this paper?" And they're like, "No." "Oh, yes, I found one. Fine."
Hey, we get listeners sending us papers all the time.
Yeah. Secretly, though, we're hoping that you beat us to the punch and that you find the papers and send them to us, because then we know you're doing really, really well. So they found lots of cool things with CD8 T cells and CD4 T cells. But of course, people want to know what's going on with B cells, because most of the vaccines we're going to make are related to B cells. So you'd like to know, are B cells, are the antibodies that we're making really important? Are the B cells defective? How good is this going to work? So, they did photocopy again, next page. It looks almost exactly the same. And if you didn't know your markers, you might think that they're all in the same thing. But they start looking at B cells, and they of course look at multiple different B cell populations. And this is one place where we could probably take a moment to talk about this. And so, again, there are naive B cells, just like there are naive T cells. They have not seen antigen. They're not yet making a lot of specific antibody, but B cells are different than T cells because they can make some antibody even before they're stimulated, right? This is the natural antibody. And they will express IgD and IgM, but you can use IgD and IgM markers and what they're expressing to sort of look at whether they've been activated or not. So once they're activated, they usually downregulate IgD and upregulate, other antibody isotypes, and they also class switch. So you can look at, where are your antibodies along the spectrum of activation? If they're if they're surface IgD positive, they're usually naive or very early after stimulation. Then they lose IgD, but they can still make IgM, and they'll make IgM at the beginning. And there's some B cells will make tons of IgM until you get some class switching going on, which means then you start to make IgG. And usually we think of the IgG as the workhorse, right? That's the one that really is good at neutralizing. And also during this whole process, which we could go into a little bit more detail if you want. But remember, these B cells are really cool in that they can also muck with the sequence of the antibodies that they make to try and make them better. So they fine tune the sequences so they can make stronger affinity antibodies. And so they're at the same time, they're class switching to make antibodies with different functions. They're also mucking with the sequence to make higher affinity. And they have to go through selection in the germinal center to make sure they're not autoreactive and so forth. But you get these class switched antibody expressing B cells. You also get memory B cells. And then we also talk about a class of B cells called plasma cells. So they basically are just antibody factories. So they've gotten their right class switch, they've gotten their right sequence, and they got the go-ahead signal, and they just turn into a factory and just make tons of rough ER, and they just churn out huge amounts of antibody per cell on a per day basis. And so those are the plasma cells. And then the precursors to that are the plasma blasts, which are proliferating a lot because they've gotten that signal, and then they're going to start to churn out the antibodies.
Yeah, and so this is sort of paralleling what we saw with the T cells in that we're able to make different subsets of the B cells that are at different stages sort of after activation, and they're having slightly different functions. With B cells, we've done, at least with the B cell subsets that are listed here, there is a little bit more of a tie to function.
Yes.
With some of these different subsets, as Cindy sort of just nicely talked through. But it's the same kind of concept that we're looking at within the B cells at cells that are at different stages of activation and sort of different stages of their life.
Right. And so there were differences, up, downs, whatever. But I think the key takeaway was there was just a massive expansion of these plasma blasts, which are the ones that have sort of gotten all their signals and they're proliferating, and then they're going to turn into these antibody factories. And so it 2/3 of them had this increased plasma blast, that, these hospitalized patients, but that means 1/3 of them didn't. Right? Which is also interesting because that might account for why there might be a proportion of the patients that are not making antibody responses. But this isn't that surprising because it's not unique to SARS-2. This is something well described for other viral infections like dengue and Ebola and so forth. You have an acute viral infection, you get a strong antibody response, you're going to have a lot of these plasma blasts circulating in the blood. So that wasn't all that surprising. They definitely saw expected features like increased proliferation of all the B cell populations. They saw clonality, which I thought was really cool because that suggested that there's antigen-specific responses happening.
So what that means is that the clonality means that there was sort of one initial B cell that was triggered when it saw antigen, and it divided and divided and divided and made lots of progeny. And so all of those B cells have the same genetic sequence. It's not as if we have that sort of random bystander activation where excess cytokines or excess other sort of immune factors are just triggering any old B cell. The clonality really tells us that these are the B cells that are specific to SARS-CoV-2.
Right, which is good. You don't want random B cell populations expanding out. You really want to put the energy into the antigen-specific ones developing. So that's good. One of the other key features they saw was a reduction in a chemokine receptor that's required for homing cells to the lymph node and B cell areas. So that's a little interesting, right? Because we want the B cells to be in the lymph node and getting the T cell help and making the antibodies and doing their class switching and affinity maturation there. But they seem to be, they have lost that marker, that homing marker, and now they're circulating around the blood. So are they lost again? I don't know. There seems to be something happening.
And that makes it tricky because to make high quality antibodies, those B cells need to interact with CD4 T cells. And if the B cells are lost and the T cells are lost, they're probably not going to be able to interact to make the best possible antibody response.
Right. The other weird thing was, you brought up the T cells, is we talked about those T follicular helper cells being really important for driving the B cell responses. One would think that if you have a whole lot of plasmablasts, you should have a really good T follicular helper response. But remember, we said that was actually down, although there was a subpopulation that was activated. So it's interesting that one would have anticipated if you have a high plasmablast, you should have high T follicular helper. And it doesn't seem to be the case. So there's some disconnect between the components of the immune response that should be working together based on everything else we know. And they don't seem to be working as we expect them to be working in this disease, whether that's driven by the infection or whether we have a magnifying glass that we can now see things that we couldn't see before, and those differences are popping out that we didn't know about before. So it's interesting. So the weird thing to me, and I'm not sure, maybe I missed something as I was reading through this, but they seemed to say there was not a good correlation between the good functional antibodies and the plasmablasts. So it seemed to me that they were saying that they got lots of these plasmablasts, but they don't seem to be making the right antibodies that they need for this infection.
Yeah. So they were specifically talking about antibodies that were binding spike there.
Right.
And so they kind of indicate, well, maybe we're getting lots of antibodies to something other than spike, or maybe there's some other things going on. Maybe, we're looking at other classes of antibodies or something like that. So they hand wave a little bit at that.
Yep.
And that caught my eye because I had been paying attention to some other literature that's been talking about spike versus other antibodies. And so the idea that perhaps they were missing some of the antibodies, maybe the plasmablasts were really correlated with the nucleoprotein antibodies or something like that, could be sort of interesting to me, at least.
Right. Yeah. I don't know. So I don't want to go into too much more details about this paper, but start to back out to the kind of the bigger picture. And that was, so they did something like, it was the first time I'd ever heard this term, the Earth Movers Distance, or the EMD. I thought, wow, that's really cool. And I can't say that I fully understand how they did it, but they just took this like hugely multi-dimensional data and just, asked it to group clonotypes or immunotypes of COVID patients. So could we actually say that some are more severe, and do those more severe ones have a specific immunotype? Can we use that to predict how they're going to, how the disease is going to progress?
Yeah. And this is sort of related to the clusters that we saw in the t-SNE plots before. But basically what they wanted to see was whether patients who had different severity of disease had enrichment or reduction in some of those fractions they made in the t-SNE plots before.
Right. And in this way, they're not just looking at one parameter at a time. So it's not just like, okay, this patient had more severe disease. Let's go back and look at this particular T cell population or this particular B cell population. But it sort of looks at it at a whole, and it asks like, what are the immune drivers that correlate with the severity of disease? So it's not just one aspect. So do they have a failure in B cell proliferation plus more of certain types of T cells, or exhausted T cells? So is there an immunotype? So is there a set of immune parameters that define a particular population of people with disease? So one of the things that they were able to do when they were doing this analysis was they took a subset of their patients and were able to take their day 0 data, but then sample them again on day 7 and see whether these parameters were stable or whether they changed. And might that indicate whether these immune parameters kind of drove those responses? And so generally what they said was that pretty much everything was stable to some degree, but there were some things like that might be expected. So the frequency of non-naive T cells went down over time. So we would expect that because you'd have more responders. And then they did things that I'm not sure I fully understand, but they used healthy donor data to determine variabilities in parameters, because one of the notorious things with human studies is there's just a huge variability. And so they used healthy donors to define variability, and then used that to actually analyze their data to get rid of the noise and see what parameters were driving these different immunotypes. And so they found that of the diseased patients, like 50% of them had an increase in the number of non-naive CD4 T cells. So that makes sense. And 30% had no change, and 20% it actually went down. So they could group these into, like, for example, just looking at CD4 T cells, 3 immunotypes, right? So some responded better. So that means they were on an upward trajectory. They were not responding well at first. They had a better T cell response later. Some were the same, and some actually went down. And so the same was true with proliferating CD8 T cells. Some of them had no change in response, and some of them went up, and some of them went down. And so there's lots of ups and downs and differences, but the question of what does it all mean in the end? And so they were able to do one more dimension of analysis. And that was, they took each one of those up downs and their immunotypes, and then they correlated that with all the different parameters that they had looked at. And they asked, is there anything that seems to change between day 0 and day 7 that is associating with driving disease? And so what they saw was that CD4 T cells going down correlated with an increase in white blood cells and neutrophils, that decreases in T cells correlated with increases in Apache scores. So what does this all mean? So this just means that if you lose your T cells, you're going to get sicker. Which now we come back to saying, how does that fit into what everybody's saying? There's some camps that are saying the disease is immune-mediated pathology and more is worse. And this is actually saying losing your T cells is worse. So it may be in this case that you need the T cells to control the disease, but yet expansion of other populations are actually driving disease. So, what the intricate little things that are coming out here, I don't know if the details are important, but just being able to take the information that we're getting and look for trends that go up or go down or correlations are important.
Maybe as you lose T cells, then you have too much virus, and then that gives you more cytokine, chemokine responses, and it's all funneling.
More immunopathology, right?
Yeah. And so they make these 3 groups, these 3 immunotypes. Group 1 that has a lot of CD4 T cell activation and proliferation, activated and exhausted CD8 cells, and a certain type of plasmablast. And those individuals seem to have increased severity. There's a group that has group 2 that has sort of less of a CD4 response, more activated and replicating B cells. And then there's group 3 who, as and I quote, an immunotype largely lacking detectable lymphocyte response, suggesting a failure of immune activation.
So which one do you want to be in? Which immunotype? 2?
In some ways, 2, but 3 seems to have pretty good outcome after disease.
Right.
And so what you'd love to be able to do is to determine whether there are differences between these 3 immunotypes and response to treatment. Is there some treatment that's better for somebody who's in type 1 versus type 3?
Right.
Or if you could somehow take this, and this makes my brain hurt a little bit because I'm imagining just how much big data analysis would have to go into this, but take this and then look at, say, genetic associations. Are there genetics that make you more likely to be in group 1? Can we say, oh, if you have this genetic polymorphism, you're going to be in group 1. We need to sort of take care of you earlier, or something like that. Being able to overlay that onto this idea of immunotypes could be really useful, because it could help us predict disease, or knowing this could help us understand better treatment options.
Oh, that sounds like it's a ways off.
Yeah.
Yes and no. I mean, they were able to at least, some of these patients had different treatments like hydrochloroquine or remdesivir. And so you could look at, do those treatments correlate with any of the immunotypes or any of the parameters that we know would be beneficial? And they didn't seem to see anything.
Right. Yeah. So
in their data set, it didn't seem to make any difference one way or the other.
So maybe if we had some other treatments that, seem to be working a little bit better, perhaps they would be more clear here.
Right. And coming back to which immunotype you want, I mean, according to their data, like Brianne said, I mean, the third group is, it seems like their response sucks, but they don't have the immunopathology and their disease is less severe. So for whatever reason, maybe they're early controllers, and so that they don't have as robust a long-term response. Then, the big question comes, do we want a vaccine that induces a robust response? Because that's the big question that everybody wants to know. So, because those that had a more robust response actually did worse.
If you mentioned that some individuals who regulate virus early, so what's regulating it? It's not a B and T cell response then, is it?
It might be more, maybe their innate immune system is working. Let's go innate immunity. Woohoo! We both really like the innate immune system. Maybe the innate immune system produces a whole boatload of type 1 interferons and innate immune responses and controls things really early before you ever have a chance to have a robust adaptive immune response.
Yeah, because the timing is consistent, because Michael Minnis said most patients who recover and don't get serious disease, the virus is down by day 7 post-symptom onset, and they don't transmit it anymore. Can't be adaptive, right? Or mostly not adaptive.
It could. I mean, by 7 days. Yeah. I mean, so everybody sort of says 7 to 14 days for antibodies, but really, I mean, that's a little long.
That's a little late.
But 3 to 5 days is when you're going to start. When I teach this, I'm like, if you've ever had the flu, you're going to start to feel a little better 3 to 5 days later, right? It's peaks and then you start to feel better. And it might, go on for a couple of weeks if flu is really bad. And I imagined it originally that this was going to be like that. But, so by 3 to 5 days, you're starting to generate B and T cells. So maybe it's just that initial response was enough to control it. So without the continuing antigen, you don't have continued tickling to make sure you get a robust response. And it just wanes.
I think that the looking at these 3 immunotypes brings me back to sort of the one caveat from the beginning of this paper, which was that most of the patients they looked at were patients with severe disease. They don't really have a huge number of, say, asymptomatic infections or things like that. And so do those patients fall into one of these 3 immunotypes, or do they have their own immunotype that is actually the one you want?
Yeah, that's really important, right? And that's because they're just capturing patients coming to the hospital, right?
Right.
As opposed to setting up a trial, which would be hard, right? Screen people. You're going to screen people and pick out the asymptomatic. Wow, it's really tough. You're going to get them all different timings and everything.
Yeah.
No, it would be really, really challenging to do. That might be a place where you do some of it in an animal model.
I totally agree, though. You need that to really say maybe there's immunotype 4, and that's the ones that clear early without any immunopathology.
Right. I mean, we would think that of those that get hospitalized, the ones that aren't having an immune response would be the worst off, right?
Right.
But apparently not.
Seems like not.
It's all about timing too, right?
Yes. So that is a caveat to their analysis. Some of this, like seeing an increase between day 0 and day 7, could just be that they crashed a little early and went to the hospital much earlier. And so they were still in the increasing, disease phase. And so we're still looking at the early acute response. So, there's a lot of questions. I mean, following these patients longer would be really cool to know, those ones that do survive and they're, out a month later or 3 months later. What do they look like if they got out of the hospital? Do they look like the recovered ones, or do you end up with a different immunotype if you had a less severe disease or a more severe disease?
I'd also love to see how they respond to other infections, right? Because I just wonder how much of this is because we're looking at these patients under a microscope, with 39 colors or whatever. We've never really done that before to this extent, right?
Right. I mean, we could also think about, maybe some of this difference has to do with what types of coronavirus infections you had previously. There's some data that T cell responses to coronaviruses cross-react.
Yeah.
This would be not really an experiment you could do, but it would be also interesting to know how these different immunotypes responded to reinfection. Were some protected more than others?
You could do a study like this with severe influenza, right? Because every year in the hospital there are a lot of people, but I bet it's never been done, right? Because
I know I saw one paper, and I did not look that up, so I can't say the name right now. But I think it was in Nature Immunology. There was a really nice paper on high dimensional analysis of patients, and it was one of the first ones I saw because we did it as a journal club, where they were able to look at the actual patient parameters, because usually what we do, right, is we don't want to deal with IRBs and all of the complications of that. So we de-identify everybody and we lose a lot of information about patients and their clinical parameters when we do our basic science analysis of, patient samples and T cell responses and B cell responses and all of that. But if you can keep that patient data associated with the immunologic data, now we can gain a lot more out of that. And I think that was the beauty of this paper, is they did that too. So you can, look at age, you can look at comorbidities, you can look at previous infections. So you can start to do that where you look at all of that previous data. The catch to that is you need enough data points, right? So one person who had type 2 diabetes or type 1 diabetes or was morbidly obese, you're not going to be able to gain any information from that. But if you've got 100 of them in a 10,000 person sample and you're doing high dimensionality, now you start to get really interesting information about what pre-existing conditions or comorbidities or whatever actually drive more or less severe disease. But we don't quite have that yet.
Yeah, it's also worth pointing out, Brianne mentioned this, you can't do this anywhere, right? You can't do this in Ithaca, New York, probably, right?
We are having more cases, but we still only have, I think the total, what they did, 149, were only a couple more than that. You'd have to have analyzed every single person who tested positive in Ithaca.
Well, and you'd have to have, the hardcore flow cytometer that can do this many parameters, and you'd have to have a really great bioinformatics team. And right. All of those patients.
We have some of that stuff. So, yeah. So one of the things I was doing in preparation for this is I started digging into, the newer papers that are coming out, because a lot of people are doing this or very similar stuff. They're doing mass spec flow, analyses, or they're combining their multidimensional flow analysis with transcriptomics. Some are doing single cell sequencing in transcriptomics, or single cell sequencing plus their flow analysis. So we're starting to get these groups of data that are much more, way more information and much more detailed. And just, if you are not in the lab right now and you have these skills to be able to start poring over these data sets, there's just so much information. This one paper in Science that has got 6 or 7 figures and some supplementary data figures, and those figures, I mean, since we're on video, I mean, this is like it's a lot of data in one figure, right? Yeah. There's just so much that they're highlighting a few things that really stuck out at them. But if you are working on something and you want to find out whether your gene of interest or your cell of interest or your pathway of interest is up, down, or regulated, you can dig into these data and find all of that information. Yeah. There's just so much.
It would be nice if they had the genotype of all these people, right? To layer that on top of it.
I'm sure people are doing it. And if you bank some cells, you can just go back and sequence, right?
For sure.
It's just they haven't done that yet. But if they've banked cells,
sure
they can do that.
Yeah. Very cool.
Yeah.
Right. Thank you. Is that
anything else?
I think that's it.
Yeah, I think that was quite a bit.
I'm exhausted.
I tried to keep it as.
What markers do I have from exhaustion? Yeah, that was great.
But, what I took away from this though is they said that in their discussion is like the most important takeaway is the immune responses are heterogeneous.
Yeah.
We knew that.
Yeah, we knew that.
We just know how heterogeneous they are on a way bigger level now. So, it was a fun paper to read. And I think it really is going to be a groundbreaker in the field of understanding what's going on immunologically.
Great. Thank you. It was great.
Yeah.
Do you guys have time for a few emails here?
I do. Sure.
Okay. We they've been piling up because we only record once a month. Brianne, why don't you take the first one?
Sure. Mary Claire writes, "Hello, immune team. Great coverage of COVID-19 immunology. Thank you. My understanding is that CoV and CoV2 have a fairly wide tropism, possibly including neurons. I've read that the immune response to a coronavirus infection in the CNS can include macrophage-mediated demyelination. Can you explain this? How and why does this happen? Thanks again. Please keep up the excellent work. There are lots of us out here listening. Cheers, Mary Claire."
Mm-hmm.
So macrophages in the brain are called microglial cells, and their main task is kind of to keep the neurons healthy. And so they prune and they clean and they do lots of things that I don't know all the details of. But when they're malfunctioning, you can end up with demyelination. I don't know a lot about that in this disease, but if that's happening, that's kind of terrifying.
Yeah, and so I also do not know much about it in this disease, but what sometimes can happen is that those macrophages can clean up some myelin and end up inducing an immune response where T cells can come in and further lead to some damage to the myelin. And so that's the very big picture way that can happen. Although, again, I don't know if that's specifically what's going on here.
Yeah, there are a lot of neurological complications in COVID-19 patients. Not at all clear if it's related to virus replication or secondary effects, right? And we're still waiting on that. You can take brain organoids and infect them, but that doesn't mean that the virus is actually infecting people's brains. So we have to wait. I asked Tony Fauci whether it was infecting multiple places. He said, we'll have to wait till autopsy results and see. Although I would say, even if you see particles on autopsy, it doesn't mean the virus is actually replicating, but you could do other things as well. Cindy, can you take the next one?
Sure. Max writes, "Hello, immune team. I have been enjoying your podcast and learning a lot. Completely hooked to immune, TWIV, etc. I have a question about an item mentioned in episode 29 when talking about antibody therapy for COVID-19, particularly use of convalescent plasma. At around time mark 1 hour and 22 minutes, there was a discussion of a phenomenon described as antibody interference. If I understood this correctly, the potential concern is that giving patients this sort of therapy may ultimately block development of endogenous, patient's own antibodies, and perhaps, not sure, other aspects of the immune response. And yes, the risk-reward calculus may still go in favor of the therapy, but I was curious about the potential issue you described. Could you clarify this, please, and if possible, provide some literature references describing this phenomenon and or examples? Searching the term antibody interference led me into a largely bioanalytical direction that is not what you were describing, I think. Since the airing of episode 29 on March 31st, the following preprint came out, and you may find it interesting. As I recall, the last author, Arturo Casadevall of Johns Hopkins, was one of the early advocates of this approach in the context of COVID-19. Though, of course, as the hosts of Immune indicated, the general idea has been around for many decades, if not a full century already. The link to the MedArchive article, timestamped May 12th, and it's included there. Thanks much in advance, Max. And he puts a disclaimer. I am not an immunologist or a virologist, but a chemistry-trained scientist in the biotechnology industry. I don't think that discounts him as a someone knowledgeable in the area in general. Do you want to take it, Brianna? Or you want me to talk about antibody interference?
Sure. So there are different antibody types, like Cindy mentioned, and typically a B cell will make IgM early in its life and switch to IgG. But IgG being present and responding to a particular antigen can actually bind to a naive B cell and inhibit that B cell. And so having some IgG around will inhibit the ability of new B cells to make a response. So if we are doing antibody therapy, we're giving you IgG, that could inhibit your B cells from making a response. And if you wanted to kind of look at some literature about this phenomenon, you could look at the clinical use of something called ROGAM, which is used in moms who have ABO blood type mismatches, and we want to stop them from making antibody responses to their baby's ABO blood type. And so it's basically giving antibodies to block mom's ability to make those antibodies themselves.
And in addition to that, I thought there was potentially also a component if we are neutralizing the virus before antigen presenting cells can actually take it up and present it, that you might also have a defect in generating T cell responses and get antibody responses.
Yeah, absolutely.
So I think there's multiple ways to interfere with the process as well.
The paper he links is a safety study of convalescent serum. It shows that it was tolerated and so forth. But since then, this was tried in very sick patients, and it really didn't help them because when you're very sick, an antiviral is really not what you need, as you've heard, right? You need to suppress your immune response. So it's not helping them. You have to give it much earlier, and it's hard to do that because you're not sick. You're not very sick, and this is an intravenously administered therapy, right? So usually you get it in the hospital. It's the same with monoclonal antibodies. It's going to be very difficult to give it to people very early. It's not like Tamiflu. You can pop a pill
right
at your first sign, but we don't have that.
Especially when you may not know that you're sick, you're probably not going to go for an IV treatment.
No,
that's right.
Not at all.
Yeah, I think it's a problem with a lot of immunotherapies and a lot of therapies in general, right? We are allowed to test them on the sickest patients because they're less likely to do more harm than good, right? But the problem is that a lot of them would only work if they were tested earlier. Yeah. And so how many of the drugs that we test fail because they don't get tested on the right population?
Right. Jody writes, I absolutely love the podcast. Thank you. Still listening to past episodes. So I apologize if this has been covered. I have an acquaintance who is most unhappy about the inconveniences of pandemic restrictions. I think most people are. She has some autoimmune issues, and as a result, she sees an immunologist who she says told her the lockdown of society is damaging our immune systems. she says there will be massive numbers of upper respiratory tract infections in the schools this fall because children haven't been allowed to be exposed to each other. Can you talk about this? Would the immune system be damaged or weakened in any way by the social restrictions we have implemented? I think it sounds unlikely, but I would never presume to know more than a physician who specializes in immunology. Why not? For a physician to say the restrictions are damaging your immune system is just absurd. Absolutely absurd, right? It's damaging our economy. It's damaging our lives, but no. All right, I'll go on. I'm sorry. What are your thoughts about non-COVID disease incidents when society resumes essentially normal operations? Are our immune systems going to be lacking diversity in response and become overwhelmed by a sudden bombardment of viruses and bacteria undiluted by time, those pathogens we may have been but weren't exposed to during the time we have been socially restricted? Well, you guys can weigh in.
So I am not a physician who specializes in immunology, but my knowledge of immunology would say that's just crazy.
Yeah.
The only thing I could say is that stress does have an impact on immune responses, and given the stress that people have been under upon exposure to maybe common cold that we weren't seeing circulate around as much, maybe that could be what they're going at. I don't know. But just generally, I don't think that anything we've done is going to negatively impact your immune system, and just the opposite argument could be made, right? That we've rested our immune system because we haven't exposed ourselves to anything. So we'll be more prepared when we find something in the fall. I think that's also crazy.
Yeah, I don't really think there's anything to this. I know that I can make sort of connections between this and the hygiene hypothesis, but that assumes like your home that you're staying in right now is 100% sterile. And I'm sad to tell you that it is really not.
Yeah.
So your immune system is definitely not going to be hurt in any way. You mentioned something about whether your immune system is going to be lacking diversity in its response. One of the coolest parts about at least the adaptive immune system is the adaptive immune system generates diversity before you are ever infected. So diversity happens much earlier in the process, and there is no chance that you are going to suddenly lack out on diversity in this response.
Nope, because your T cells are chosen and done before, like right after birth, and then your B cells have the ability to continuously adapt. So none of that makes any sense to me.
Hey, you'd have to live in a bubble, right, to not get any infections of any sort. It's crazy. Whenever I hear of doctors saying things like that, it really drives you crazy, and realize that not everyone is educated equally, even in medical schools, right?
Yeah, those sort of things really frustrate me a lot. I think that these sorts of things are one of the reasons why I started putting my immunology lectures online, just so that I could have something out there. I could feel like I had done something.
Yeah, no, I agree. I'd do the same. It's just that we don't reach enough people, apparently. All right. We have plenty more, but we'll get to them next time. Next
time we'll choose not quite as dense a paper, and then we can do more email.
Sure. That's Immune 33. You can find the show notes at microbe.tv/immune, or you can listen on, a podcast player. If you do, please subscribe so you get every episode. It's just once a month. And if you have questions or comments, immune@microbe.tv. If you like what we do, go over to microbe.tv/contribute, and there are a couple of ways where you could contribute some funds to help us out. Cindy Leifer is at Cornell University, at Cindy Leifer on the Twitter. Thanks, Cindy.
Thank you.
Good to see you guys again. Brianne Barker's at Drew University, BioProf Barker on the Twitter. Thanks, Brianne.
Thanks. It was great to be here.
And I am Vincent Racaniello. You can find me at virology.blog. Music on Immune is by Steve Neal. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.